Hello everyone, welcome to the GATE NPTEL course, and the topic today is cell structure and signal transduction. The question came in the year 2016 in biochemistry section, and this question had 2 marks. So what this question is stating, the sequence of a polypeptide that forms a transmembrane helix is shown below. So they have given a sequence of amino acid of a transmembrane helix. Okay, and they have marked the numbers of the sequence number is given also. Which one of the following segments of the peptide is most likely to span the membrane? So there are 4 options. So now first you need to know that what is the property of your transmembrane helix. So let us first look into that how what determines that which helix will be actually embedded into the transmembrane section or embedded into the phospholipid bilayer. So after doing this concept, then we can actually come back and solve this question. So if you see here that your lipid molecule, they have a hydrophilic head and a hydrophobic head, a hydrophobic tail, hydrophilic head and a hydrophobic tail. So this hydrophilic tail, they actually points outwards, and the hydrophobic tail, they actually goes inside the membrane to form the lipid bilayer. Okay, so this protein, if you imagine this protein, this section which is getting embedding into this hydrophobic layer of the lipid bilayer, then this section should be highly hydrophobic or arranged with non-polar amino acids. So if this section is hydrophilic or charged in nature, then they cannot be attached or they cannot go through this hydrophobic phospholipid bilayer. So what is the first criteria here is that this section should be hydrophobic or non-polar in nature, non-polar uncharged. So these transmembrane proteins which are amphiphilic in nature, amphiphilic means they are both hydrophobic and hydrophilic, like this lipid moieties, like this has a hydrophilic head and a hydrophobic tail. So similar to this, these are amphiphilic molecules. Similar to this, transmembrane proteins are normally amphiphilic in nature. This section, the extracellular section and this cytosolic part, they are mostly hydrophilic in nature. But the transmembrane part or the section which actually goes across the membrane, that is more hydrophobic, and non-polar uncharged amino acids are arranged in that section. So that's why these molecules should be amphiphilic in nature. The hydrophobic domain passes through the membrane bilayer and covalently interacts with the interior of the bilayer, which consists of hydrophobic group, and the other end is hydrophilic, which is exposed on the opposite side of the membrane bilayer, as I discussed. So this section, if you see my cursor, this is more hydrophobic, that is inside this lipid bilayer, and this transmembrane section of the protein is actually interacting with this hydrophobic group. For that, this should be uncharged or more hydrophobic in nature. Whereas the two membrane exposed, this is the ectodomain and this is the cytosolic part. So these are exposed to the solvent. So this cannot be hydrophobic, and this needs to take part in several interactions with other proteins. So these are mostly hydrophilic, and these inside the cell also they are hydrophilic, but in between them, if it is a membrane protein, then the transmembrane section is more hydrophobic and uncharged in nature. Both domains are divided by the transmembrane proteins segment of the polypeptide chain spanning the membrane, which largely contains non-polar amino acids, which cause peptide bonds are polar. So all peptide bonds in bilayer create hydrogen bonds with each other in scarcity of water. In alpha helix form, maximum hydrogen bonds are formed between peptide bonds by transversing the segment of transmembrane protein spanning the membrane bilayer. So what is happening here? These peptide bonds are actually polar in nature, so that this will not be suitable to be present in this lipid bilayer. For that, what they do is that this peptide bond, they form hydrogen bonds and they form helix like structure, where the side chains of the side chains are actually exposed to the environment, whereas the peptide bond is actually inside this helix. So that's why these transmembrane regions are consist of mostly helical regions, like as you see in this picture. So now what is the criteria? It should be more hydrophobic or it should be uncharged and non-polar in nature. Those amino acids which contains this nature, then what we can say that they are actually going to present in this transmembrane region. Okay. Now if you go and see this question, note down the sequence first, then we will be able to solve it. So the answer to the question is the cell membrane is made up of lipid, hence it is non-polar membrane. As we know, non-polar attracts non-polar, so we should have sequence that has more non-polar amino acids, as I explained you just before this slide. So for if I take the first option and check that how many polar and non-polar amino acids are present, let us count. So this is the arrows that you see, those are the non-polar amino acids. So total there are 20 amino acids which are given, 20 amino acid sequence is given, 20 amino acids are present in that sequence. Among them, in the first option, if you count, there are 9 polar but 11 non-polar amino acids. So the sequence which will have more number of non-polar amino acids, that will be our option, because that will be more suitable for embedding into the hydrophobic layer. And in the second option, if you count again the number of polar and non-polar amino acids, there are 8 polar and 14 non-polar amino acids present, which is greater than the option A. So now for us, the option B is suitable, but this is not the correct. Let us check the other two options. So in the C option, if you count, there are 16 non-polar amino acids greater than both A and B. And in the D option, if you count, it is 15 non-polar amino acid. So if you compare four options, the third option or the option C has the most amount of greater number of non-polar amino acids, and there are three only polar amino acids present in option C. So from here you can actually say the answer to this question is option C. For your reference, you can go through these two courses of NPTEL. The first one is Introduction to Cell Biology by Professor Nagaraj Balasubramaniam and Professor Girish Ratnaparkhi of IISER Pune. And the second course is of Cell Biology by Dr. Sanjukta Patra of IIT Guwahati. You can go through these two courses to clear your concepts in cell biology.